Hello everyone, today we will analyze the insulin receptor INSR. As a key member of the tyrosine kinase receptor family, it is a vital membrane protein mediating insulin signal transduction, and its functions are closely related to the body's metabolic homeostasis and the development of various major diseases. We will elaborate on what INSR is, its expression and structural distribution, signaling pathways, functions, diseases, and target therapy. INSR belongs to the receptor tyrosine kinase family and is the core membrane protein mediating insulin signal transduction. It is mainly expressed in the core metabolic target tissues, such as the liver, skeletal muscle, and adipose tissue, and is also widely distributed in various sites, including peripheral nerves, kidneys, and immune cells. This reflects insulin's core role in the body's metabolism, cell growth, and even tumorigenesis. INSR's physiological functions cover multiple aspects. As the core of metabolic regulation, it binds to insulin to activate downstream signaling pathways, promoting glucose uptake, and glycogen and protein synthesis to maintain blood glucose stability. It also participates in cell proliferation and differentiation, regulating the development of tissues such as the embryonic pituitary gland. In the brain, it is expressed in neural stem cells, affects neuron regeneration, and is associated with neurological diseases, such as Alzheimer's disease. In adipocytes, it can specifically inhibit lipolysis and regulate lipid metabolism. Encoded by the INSR gene, INSR is a type I transmembrane protein, composed of 1,382 amino acids with a molecular weight of about 156 kDa. Its precursor protein is hydrolyzed into IN chains, forming an heterotetramer via disulfide bonds. It has specific insulin-binding sites distributed on the chains and the junction of fibronectin domains. The chain is the functional core with tyrosine protein kinase activity, and the intracellular ATP binding and autophosphorylation sites are the key to downstream signal activation. INSR exerts its effects mainly through two core signaling pathways. First is the Pi3K oct metabolic pathway. Activated through steps such as IRS phosphorylation, it can promote glucose uptake, inhibit apoptosis, and regulate glucose and lipid metabolism. Second is the RAS-MAP pathway, which mainly regulates cell growth and differentiation and is also associated with tumors. For example, glioblastoma relies on this pathway for cell survival. INSR dysfunction can cause a variety of diseases. The most common one is type 2 diabetes, which leads to hyperglycemia due to insulin resistance and accounts for more than 90% of diabetic patients worldwide. INSR gene mutations can cause severe hereditary diabetes, such as type A insulin resistance syndrome and Donahue syndrome, resulting in severe metabolic abnormalities and developmental problems. Type B insulin resistance syndrome is caused by anti-INSR antibodies blocking signal transduction, leading to alternating high and low blood glucose. In addition, insulin resistance can trigger metabolic syndrome, and impaired INSR signaling in the brain can exacerbate Alzheimer's disease. The research and development of INSR-targeted drugs has achieved remarkable results, and a number of INSR agonists have been approved for marketing in China. Such drugs treat type 2 diabetes by binding to INSR to activate downstream signaling pathways and mimic the effects of insulin. Among them, two types of insulin deglutic developed by Jilin Huisheng Biopharmaceutical Co., Ltd., were approved on July 30th and August 5, 2024, respectively. The insulin glargine biosimilar developed by Hafeiti and My Biotechnology Development Co., Ltd., was also approved for marketing on June 18, 2024. To sum up, INSR is a key membrane protein for insulin signal transduction, widely expressed throughout the body. It regulates glucose and lipid metabolism, cell growth, neurological functions, etc., maintaining the body's physiological homeostasis. With the heterotetramer as its structural basis, it transduces signals through two core pathways. Its dysfunction is associated with various diseases such as diabetes, autoimmune diseases, and tumors. At present, a number of INSR agonists have been approved in China, providing effective targeted therapeutic options for type 2 diabetes. and its therapeutic potential for more diseases remains to be further explored. More targets sharing. See you next time.